Hey guys, hi, welcome to Unacademy. This channel is Unacademy Future Doctors, and I'm your educator for biochemistry, Dr. Shivangi Shukla. Let me just check if the live stream has started or not. Do let me know in the chat section if I'm visible and audible to everyone. Just check if the live stream. Yeah, all right. Okay, so we are meeting after a break, after the holy break, isn't it? How was your Holi, everyone? How did you celebrate Holi? Do let me know in the chat section. It's I have always maintained it is as essential to play as it is to work, because all work and no play makes Jack a dull boy, isn't it? And at the same time, all play and no work will make him a mere toy. So we have to have a great balance, and that is what I have been a proponent of ever since we've been together here on an academy. So, keeping this in view, let us quickly start today's session. We have a clinical application based session, which we have been continuing the application part of from the chapter of nucleic acids. So, last time we read about RT-PCR and all. We also have to have a class on the CRISPR-Cas9 technology, which is the latest technology in the field of molecular biology. So that is also an applied concept from biochemistry that you should know as a student of biochemistry. Okay, today we'll be discussing about translation, all right, and the applied concept, the concept of fidelity. What are the three aspects of translational fidelity? Okay, all these things we're going to discuss in today's session. Okay, so let us continue. About the NEET PG subscription for Unacademy. I'll just brief you about this. All the newcomers, the iconic as well as the plus subscription are the two options available for you to choose from. Okay, both of these will provide you access to video resources from top educators across the country. You'll have regular classes. You will have a timetable according to which you can discipline your studies. So it is a great mediator of these subscriptions. They don't just provide you with study material or doubt solving classes or quizzes. Rather, they will instill a sense of discipline in you. Because if you see the I have been the greatest supporter of self study. Okay, and all of you know the best and the most effective method is to study by yourself. But you lack experience in this field because you haven't yet taken the exams, or you have taken the exams once or twice and you require more experience. So we as experienced people, we who have taken the exams, got through, have completed the studies, now we are here to guide you through this. The most important fact in preparation, in any type of preparation, is to know what to do and what not to do, to filter, to skim through the important, to filter out the important stuff from the unimportant shafts. Okay, so this is what this subscription provides you with, and it is the most inexpensive in its league. Also, you will not you will or the classes that are provided here on the Unacademy app simulate the physical classroom environment, because there are a lot of features, state of the art features, like a raise a hand feature, ask your doubt, polls, interactive chat sections through which they can have one-on-one discussion with the students. Also, Unacademy has come up with open house sessions. These open house sessions are 1:1 audio sessions between the educator and the learner, through which they can chat and the learner can ask anything, any doubt the learner can ask of the educator. It will be as if you're talking to your mentor over phone. Okay, so it is a very interactive and a novel concept that has been introduced by Unacademy. All right, so yeah, there are a lot of revision sessions going on for the NEET PG examination on May 21st. Okay, also there are revised rates for NEET PG subscription. So please, before there is a hike in prices, do avail of this, of the current prices, and do take the subscription, because guys, according to me, it is the prices are peanuts, because we end up spending way more in 1 month on food, shopping, and whatnot. Okay, so this is really inexpensive, and you can get additional 10% discount if you apply the educator code DRSHUKLA Dr. Shukla while checking out. Okay, so the 10% educator discount is available if you apply the code DRSHUKLA. All right. So now I'll just brief you about the different levels of translation and the whole process of translation, and then we'll move on to the aspect of fidelity. Fidelity also, as we go through, I'll be explaining you about that. And the diseases on translational errors and all we have already discussed. There are a lot of protein misfolding. There are not just misfolding. There are a lot of translational. There are a lot of diseases that occur due to translational errors, like Alzheimer's, like ADHD. Okay, a lot of inheritable diseases are due to errors in translation. Okay, so for understanding errors in translation, the etiology part we can write about errors in translation. But what is it? What is translation? Translation, and at what level does the error occur? This is what we should have a fair idea about. Okay, we're not going to go into much detail because that is outside the scope of what we have to study. But at least we should know the basics. All right. So, what is translation? Translation in common parlance, translation is converting one language to the other. Similarly, translation in nucleic in nuclear biology or molecular biology parlance is conversion of the language of mRNA into proteins. The language of mRNA is that of nucleotides. Okay, is that of codons, the genetic code. Now you convert that into the language of proteins. And what is the language of proteins? The language of proteins is amino acids. So you convert what is stored or what is meant by the genetic code into amino acid. Now both of these will mean the same, just like one language to the other. Now if I'm saying my name is Shivangi in English and I translate it into Hindi saying Mera naam Shivangi hai, both of these mean the same. It is just that the same information in English has been translated into Hindi. While both mean the same, both carry the same information. Similarly, the information carried by the genetic code is converted into amino acids and both mean the same. That means both will be meant for a particular body function. Understood? So this is what is translation in simple format, in simple terms. Okay, so now I've seen a lot of students getting confused as to what is translation, what is meant by translation. Now they get confused between transcription and translation. Okay, so translation, now again, you should always correlate it with the normal English words. Now what is transcript? Transcript is any short form or any part of a huge document that carries the information, that carries the summarizing information from that document. Similarly, the huge document is our DNA that contains introns, exons, that contains a lot of intervening segments. Now, out of those, if you take out the important part, that is the genetic code, that is mRNA. Now, that is the summary, that is the transcript, that is the part. Understood? So transcription is that. Now translation means conversion from one language to the other. Conversion from mRNA to protein to amino acids. Okay. So always try and remember the English meaning, and then you can apply it here in nuclear medicine, or not nuclear medicine, molecular biology. Understood this or not? Okay. Do not get confused with these simple questions. Now, information in the sequence of mRNA is translated into the sequence of amino acids in the proteins through the process of translation. Alright, now mRNA molecules and amino acids, both of them have no affinity for each other. It is not like a particular mRNA, say suppose it is AUG. This mRNA containing the sequence AUG, it is not that it will go and it by itself go and get near or stand near or attach to methionine. No, both of them don't know each other. Okay, now they will have to have an adapter molecule. That is a translator. Now suppose I am a purely Hindi speaking person and I go and stand near a Spanish speaking person. Okay, now both of us have to communicate with each other, but we don't have any common words in between, or we don't know how to start because none of us understands the language of the other. So we need to have a translator. And what is the translator or the mediator between us? It will be another person. Okay, through who will understand both Spanish as well as Hindi. Similarly, this translator or mediator or adapter molecule between mRNA and amino acid is tRNA. So this tRNA is going to associate both with mRNA as well as with amino acids, just like the translator who will understand both Hindi as well as Spanish. Understanding this, it is very important for you guys to always understand by example so that you remember it for life. Okay. Now, what about this triplet codon? What is this brouhaha about triplet codon? Alright, let me explain this to you. How many nucleotides do you know of? All of us know about 4 nucleotides or 4 nitrogenous bases, whatever. That is A, T, C and G. Now this T can be substituted with U in RNA. Okay. Now these 4 can be arranged in different sequences to code for amino acids. Okay, now if suppose one amino acid is coded for by one nucleotide, how many different types of codes can you have? There are 4 nucleotides, so 4 nucleotides can and 1 has to be used at a time. So 4 to the power 1, that is 4. There can be 4 codes. Now if you take 2 nucleotides at a time for a code, then 4 nucleotides can be and you take 2 at a time, so it will be 4 to the power 2, that is 16 codes. But how many amino acids do you have? You have 20 amino acids. So still these codes will be insufficient to code for this 20 amino acids. Now let us take 3. Okay, 4 to the power 3 is 64. 4 into 4 into 4. There can be 64 codes or codons. Okay. Now this much is sufficient for 20 amino acids, even more than sufficient. Okay. So what did we get? We got a triplet codon, that is 3 nucleotides will code for one amino acid. A 3 nucleotide code will code for an amino acid. A 3-letter unique code for each amino acid. Okay. And these 3 letters stand for what? They are different nitrogenous bases or nucleotides. Okay, like AUG, UUU, AUC, UCU. Okay, all these. Alright. Now, one more thing you have to notice here is the fact that there are, if you have a triplet codon, which you do in nature, then there are 64 codons that are available to code for 20 amino acids. बहुत नाइंसाफी है, isn't it? बहुत नाइंसाफी है. You have 64 whole codons and just 20 amino acids. So these 4 codons, more than one codon will be clamoring for one amino acid, isn't it? 64 codons have to code for just 20 amino acids. So one amino acid will have more than one codon. Okay, one amino acid can be coded for by more than one codon. Okay, but one codon that is coding for a particular amino acid, that codon can mean only one amino acid. That codon cannot stand for any other amino acid. Do let me know if you have understood this. This is what these two things are what constitute the first two important aspects of the genetic code. Very, very important, guys. Okay, degeneracy of genetic code. A degeneracy of the genetic code is the fact that more than one codon can code for one amino acid. Now, for those of you who are very observant, they will see I have written 61 here and 64 here. Where are the three? Where are the rest of the three codons? Why have I not included them here? Any of you, can you guys tell me in the chat section, where are the rest of the three codons? The rest of the three codons, guys, the rest of the three codons are, what are these? They are stop codons. What are these? They are stop codons. Okay, what are these stop codons? Amber, ochre, and opal. Okay, amber, ochre, and opal. Now it is your homework to tell me what is the triplet codon for these three. I have discussed this time and again. Now you have to tell. Okay, all right. All right. Now, what is degeneracy? Degeneracy is the fact that more than one codon can code for one amino acid. What is unambiguity? Unambiguity is the fact that one particular codon will code for only one amino acid. This is unambiguity. One particular codon will code for only one amino acid. What is non-overlapping? Non-overlapping means that the codons are read without overlap. Okay, like suppose the genetic code is AUG, Okay, now you cannot read it as AUG and then you cannot read it as AU and then GUC, then CUC, then UCU, CUU. You cannot overlap different codes together. Understood? Without any overlap. So it is AUG, UCU, CUU, CCA, GUG. This is how it is read. Not as AUG and then UGU and then again GUC. You cannot overlap different nucleotides. Alright. Now you will say it is just a triplet codon. So you can overlap triplets you can read them as triplets anyhow. No. You don't have to overlap. Alright. Excuse me. Next. Non-punctuated. Okay, the genetic code is non-punctuated. Uh, there is no break in between until you enquire what? Until you encounter what? Until you encounter one of the three stop codons. Okay, UAG, UAA, UGA. These are the three stop codons. So your homework also, I have given you a hint. You just have to match the column. UAG, UAA and UGA. You just have to match these three with these three. That is the homework. Now your homework I have made simpler. Okay, understood? Now the last characteristic of the genetic code is that it is universal. Okay, universal means that this particular code, that is AUG, will mean methionine in you, in me, in every other person. Okay, it is the same for every other human, every other Homo sapien. Okay, this is what is the genetic code. But this universality has one exception, that is between the mitochondria and the cytosol. Now, all of you would know that the mitochondria also contains DNA, that is the circular DNA. Okay, now that DNA present in the mitochondria will also have some genetic code, and that mitochondrial DNA is transmitted from the mother to the child only. Why? Because the mitochondria in the sperm is left behind. It is not fertilized. Okay, so all the mitochondrial inheritance diseases are transferred only through maternal inheritance. Okay, understood? Now, having said that, the mitochondrial DNA, the genetic code contained in the mitochondrial DNA, and the DNA that is translated in the cytosol, the nuclear DNA that is translated in the cytoplasm. There are a few differences between the genetic code present in the mitochondrial DNA and the cytopholic DNA. What are these? Now, the codon AUA codes for isoleucine in the cytoplasm and methionine in the mitochondria. Whereas what is methionine in the cytoplasm? Now this is again your homework, although we'll be reading about it later on in this session only. But you should be you should be careful about that. You should be alert, and I need the answer in the chat section for that. Okay. So AUA codes for methionine in the mitochondria, whereas it codes for isoleucine in the cytoplasm. But so this is a breach of the rule of universality, because this AUA is not universally isoleucine, or it is not universally methionine. In the mitochondria, it is something else. In the cytoplasm, it is something else. But keeping in loyalty with the rule of universality, this AUA will be isoleucine in the cytoplasm of all Homo sapiens, and it will mean methionine in the mitochondrial DNA of all Homo sapiens. So in that way, universality is maintained, but between the cytoplasm and mitochondria, there are a few differences. Now again, AGA and AGG, they will code for arginine. Arginine is a semi-essential amino acid. They will code for arginine in the cytoplasm, the translation of mRNA in the cytoplasm, and they will act as stop codons for mitochondrial DNA translation. Now, what are stop codons in cytoplasm? UAG, UAA, and UGA. These are the stop codons in cytoplasm. They will act as stop codons in the cytoplasm of all Homo sapiens. Whereas AGA and AGG will act as stop codons for mitochondrial DNA of all humans. And AGA, AGG will act as arginine, will translate as arginine in the cytoplasm of all humans. Understood or not? Okay. Now, if there are any questions till now, do let me know. Any questions till now? No questions? So shall I continue? Okay. Yeah. So this was about the genetic code. Next, we'll read about a few post-transcriptional modifications. Now, they are very important, very, very important, because they remove the unnecessary stuff from the mRNA, and only the stuff that is to be incorporated into proteins is left behind. Okay, so the one important post-transcriptional modification is splicing. What is splicing? Splicing is removal of the intervening segments or introns, intervening segments from removal of intervening segments from exons. Okay, the mRNA that carries the genetic code. These intervening segments they are junk. They don't have any information that is of use in the protein translation. Okay, so the one requisite for the process of splicing is the spliceosome. What is the spliceosome? Zome is any body, and splicio means splicio is something that is related to splicing. So the primary RNA transcript, that is the raw RNA that has freshly come out of transcription. This primary RNA transcript will associate with small nuclear RNAs. U1, U2, U4, U5, U6. These 5 small nuclear RNAs, they will associate with the primary RNA transcript. Along these 3 will make the spliceosomes. And how will the spliceosome carry out splicing? The exon, so see, suppose this is the intron, this has to be removed. This intron exists between the exon 1 and the exon 2 and it has to be removed. Now how will it be removed? The splicing will move from the 5' to the 3' end, just like any other activity in the cell. Okay, just like any other process, just like DNA replication and transcription and translation. The splicing will also move from the 5' to the 3' end only. So how will it move? This is the primary transcript. Now this has to be removed. Now how will it happen? It will happen by, there will be first a nucleophilic attack at the 5' end of the intron. It will be removed like this. And now this intron will be folded over. There will be a lariat or a latch formation. Okay. Now this will folded over. This will be. Okay. And then there will be an attack here at the 3' end also. Then this will be removed like this. It will be removed. And the 2 exons will be ligated together. Okay, so now this whole segment is useful. This whole of this segment is now used for translation. Okay, so this is what is splicing. Okay, what are the other post-transcriptional modifications? Can you guys name them? One is capping. The other is tailing. Now, very briefly, can you guys define capping and tailing? Capping is addition of methyl guanosyl cap at the 5' end of the primary RNA transcript. And what is tailing? Tailing is addition of poly A tail at the 3' end of the primary RNA transcript. So even if you don't know the definitions, you should just know the keywords. By knowing the keywords, you will be able to put in the correct definition, because all the articles, conjunctions, prepositions, they are not necessary for the definition. For the definition, what you should know are the keywords, and then you can frame your own sentence. Okay, so capping, tailing, splicing, 3 important post-transcriptional modifications which I expect all of you to know the basics of. Now, one very important thing, this is the methyl guanosyl cap. Okay, this is a special type of nucleotide. Guanosyl is what? Containing G, guanosine. But methylated guanosyl, special type of nucleotide. Now, where is this methyl group coming from? Can any of you tell me where is this methyl group in the methyl guanosyl cap? Where is this methyl group coming from? Anyone in the chat section, can you guys tell me where is this methyl group coming from? Come on, I'm waiting, come on. Tell me, anyone, where is this methyl group coming from? No one is venturing. No one is venturing an answer here, is it? So this methyl group, this is going to come from S-adenosyl methionine. And what is this S-adenosyl methionine? This S-adenosyl methionine, guys, is the universal methyl donor in the body. It is the universal methyl donor in the body. And it is involved in methionine metabolism, about which I'm going to tell you in another session, methionine metabolism. Okay, it is very, very important, guys, how methionine converts to homocysteine, which will then convert to cysteine, involving S-adenosyl methionine, S-adenosyl homocysteine, and the one carbon transfer mechanism involving vitamin B12 and tetrahydrofolate. All those who know this can tell me in the comment section. Okay, do you guys know this? It forms the basis of most of the important reactions of biochemistry. Okay, and you'll get a lot of questions on this. At least one question in your professional exam or pre-professional exam is guaranteed from one carbon transfer reactions, folate metabolism, all these. Okay, very, very important. So I think we'll break here. Then we'll resume in another session. Tomorrow I'll take up the other session, okay, and we'll resume from here. Stay tuned for the 7 p.m. session of image of the day and the 10 p.m. session of how to resume studies after a break. Okay, so take a break for 15 minutes and we'll meet at 7 p.m. Okay, with an interesting session of image of the day. Okay, guys, then see you. Bye bye. Take care, everyone. If there's any doubt, do let me know. I'll stay for 1 minute. If there's any doubt, do let me know in the chat section, okay? I'll wait for your doubts, guys. And do revise this, whatever we are reading. I hope you guys revise, keep on revising, because then what happens is once you lose touch, then you tend to forget what you have been reading. Okay, so it is very, very important that you keep on revising it timely. Okay. Any doubt, do let me know about till where we have read tonight. Any doubt? And I want all of you to appear for the image of the day session at 7 p.m. Okay. I'll give you 5 minutes to look at the image and then answer the questions that follow, and then we'll have a discussion. Okay. Now, NEET PG is coming up in May, so you guys need to be very thorough with the revision. And all the MBBS first prof students, most of you would have been done with your professional examinations. But those who are yet to take the examinations would be round the corner for many. Also, all the new entrants, all the new all the people who've got who are newly admitted to the first professional exam, to the first year of MBBS, they also need to keep up with this, because side by side, even because what happens is, just when you enter MBBS, you have taken a very major entrance test, that is the pre-medical entrance test, and then you are in no mood to study this heavy duty stuff. So that is why it will be preferable by many to just watch videos, okay, as some light reading. Therefore, I advise you guys, if you're not interested in the heavy duty stuff of attending lectures and reading by yourself, in these lectures, these sessions, these video sessions are very, very helpful to break the inertia and get you into the momentum of study. Okay, also to solve all your doubts. So please follow these video sessions. Okay, they're hardly half an hour sessions, guys. And including doubts, including notes, including diagrams. So this is how, see, notes are directed at how you should answer the questions in the. So make good use of these, guys, because we are here only for you guys. Okay, understood then? Okay, since no doubts are coming up, I'll be quickly concluding this session so that I can smoothly transition to the next one at 7 p.m. Okay, so all of you do stay tuned for the 7 p.m. session and please join in and answer. Even if you're not sure of the answer, I want maximum participation from all of you who are present. Okay? Alright then, do subscribe to the channel for latest updates, notifications, timely notifications. The link for the Telegram group is available. The link for my profile on Unacademy is available. You can follow me there on the Unacademy app. Okay guys, then bye-bye. Take care. See you.